is to explain in general terms how the polarity of epithelial cells enables them to absorb or secrete materials, okay? And let's kind of take a second here and describe what epithelium is. So epithelia or epithelium is really the tissues that line all of our internal body cavities. It forms the skin. So the skin is an example of an epithelial layer. It forms the lining of blood vessels. So our endothelial cells are a specific type of epithelium layer that lines blood vessels. It forms the lining of our internal GI tract. It forms the lining of the respiratory tract, the urinary tract, the reproductive tract. All of our internal body cavities, including vessels and the external skin, those lining or those barrier membranes are what we call epithelium, okay? And so these are really the surfaces across which we have transport, right? Think about the gut. Think about movement of glucose, movement of other absorbed nutrients into our bloodstream. They have to make their way across that epithelial membrane. Same thing in the kidneys. When we absorb and excrete water through the kidneys, those things, those molecules and water have to make their way across these epithelial membranes or these barrier membranes. And so transport across cells happens via the transport across epithelial cells. And this involves the movement of solutes and molecules and particles across both surfaces of our epithelial membranes. So they're going to have to make their way across the apical surface, get inside the cell first, and then they make their way across the basolateral surface, and then they can get into the bloodstream. Okay. Now, cells have several different structures. These epithelial cells have several different structures. They've got tight junctions, and tight junctions are the connections between one cell and the other cell as in terms of the lateral surface of those cells. So in between two epithelial cells, there's that tight junction, and the purpose of the tight junction is really to prevent any paracellular transport. We want anything that moves through this membrane to go through the apical surface, through the basolateral surface, and then enter the underlying blood capillaries here. And so we do not want any paracellular transport. This is what we would call leakiness, right? If you think about, if you've heard about the term leaky gut, this is what's happening. There's movement of particles that it's not going through the cell. It's actually leaking in between the middle of the lateral aspects of those cells. And so to avoid that shouldn't happen normally, we have these very secure tight junctions in between the lateral aspects of our epithelial cells. We've also got the apical surface of the cell, and then we've got the basolateral surface of the cell as well. Now, as far as the basolateral membrane or the basolateral surface, we've got a couple things located here. We've got the sodium-potassium pump. We spoke a lot about the sodium-potassium pump in our last class, right? It's an active means of transporting sodium out of the cell and potassium into the cell using the energy that is hydrolyzed when we cleave that phosphate from ATP. Also on the basolateral surface, we have potassium channels, right? We still have to have potassium leaking out of the cell, right? Remember, according to its gradient, potassium is going to be higher concentration inside the cell, lower concentration outside the cell. And so potassium is going to leak out of the cell according to its gradient via these potassium channels. We also have glucose carriers. So here we can see some of those glucose carriers. So remember those transporters that we talked about that help with facilitated diffusion of glucose to be uptaken into the blood and be circulated to our cells. This glucose carrier is located on the basolateral surface of the cell as well, okay? And so remember, facilitated diffusion, which is the example here, is also occurring by passive transport. In other words, according to that glucose gradient. And so students often confuse the movement of glucose through secondary active transport across the apical surface with the movement of glucose via facilitated diffusion across the basolateral surface. So again, be thinking about the concentration gradient of glucose. So when glucose gets into the cell, it does so actively, right? There's more glucose in the cell, there's less glucose in the lumen, right, that's being absorbed. And so because we have a high concentration here, lower concentration here, the movement of glucose across the apical surface into the cell happens actively, right? It's going to require the energy of sodium leaking into the cell according to its gradient, okay? And so when glucose moves now across the basolateral surface and into the blood where it can be circulated and uptaken by our other cells, it does so via facilitated diffusion, which is passive movement. Okay, so it's down the gradient because there's more glucose in the cell, less glucose out here in the interstitial fluid. We now have a gradient where glucose can move passively through that basolateral membrane and into the blood. Okay. Let's look at structures on the apical membrane of the epithelial cell. We have sodium channels. So again, leak channels where sodium can leak back into the cell. So again, we have a higher concentration of sodium outside of the cell, lower concentration of sodium inside of the cell. And that is facilitated by the sodium potassium pump here, right? It's pumping sodium out. And so sodium can naturally leak back into the cell according to that gradient, okay? And this is really highlighting that pump leak system that we talked about last time. So the sodium potassium pump working in concert with the leak channels is what maintains those relative ion concentrations, more sodium outside the cell, more potassium inside the cell. And so that is facilitated by the bidirectional movement through the leak channels as well as the sodium potassium pump. All right. Okay, so I think I just kind of explained all of this here. So a bit redundant here on this slide, right? The movement of sodium happening because of that natural gradient of sodium. And so sodium wants to get into the cell passively, and then it's pumped out of the cell actively because of the sodium potassium pump. Okay. Here again, same thing that we just kind of described earlier. So I kind of jumped ahead of myself there, but sodium can facilitate the movement of glucose into the cell. So the energy that is used to move glucose actively is actually liberated from sodium moving passively. So we can see how this is all kind of feeding into each other, right? Everything is kind of working to create these gradients for one another that help to keep these ions and molecules moving in their relative directions, right? Really important to understand how this is all kind of working together to bring about the absorption of important things like glucose, but then also the relative ion concentrations of sodium and potassium. Okay. All right. And I kind of answered this question earlier. So this kind of teaser question here. In this scenario, where is glucose concentration higher, extracellular fluid or intracellular fluid? And so based upon what we just described here, glucose is highest inside the cell, right? This creates that gradient for it to be moving actively into the cell across the apical surface, but then also moving passively out of the cell across the basolateral surface. All right. Again, I'm going to pause a little bit and see how well we did with that. And then we're going to get into how water can be moved across some of these membranes. Okay. And so what we'll speak about now is how water moves across our epithelial membranes. So really tying a couple of things that we've already talked about together. One is going to be that osmotic pressure that is exerted if we have more solutes dissolved in a solution. So remember our scenario that we described previously in this packet, that if I have two solutions, let's actually illustrate this here. If I've got a container here, or a cell rather, let's say we've got a cell, and I've got higher solute concentration outside my cell, and less solutes dissolve inside my cell, I now have an osmotic pull that's being created in the solution that has more solutes, right? So there's an osmotic pull being exerted in this region, in the extracellular fluid. And we can capitalize on that osmotic pressure. And this is how we move water across our membranes. We pretty much pump solutes into the extracellular or interstitial fluid here. So we have a high solute concentration or a low water concentration in the interstitial fluid around our cells. And then inside our cells, we have a low solute concentration and a high water concentration. And so we just created a gradient by simply pumping solutes into this interstitial fluid space. And so we've now created the gradient where water can flow from high water concentration in the lumen right across the cell to low water concentration, which is in our interstitial fluid space, okay? So by pumping solutes into the interstitial fluid, we create a gradient that allows water to flow according to osmosis or according to its gradient. And this is how we absorb water even across the gut, right? And across some of our other surfaces, such as the kidneys and so on. But water moves from high concentration in the lumen of our gut to low water concentration, where we've now pumped these solutes here to create that lower water concentration because we have more solutes in this space. All right, I really hope that's clear. So a question here, why is it flowing out of the cell from low to high concentration? So it's flowing out of the cell. So on the right, water itself is flowing from high water concentration. Remember, there's more water in our lumen, the lumen of our gut, to low water concentration. So the area here, the interstitial fluid that has more solutes is the area that has more of a solute concentration, but less of a water concentration. And so water can flow naturally right through the cell to this region. Okay, remember, water doesn't need any carriers, really important to understand here. Water doesn't need any mechanism, any facilitated diffusion. It's going to move from high to low right through the cell, okay? Doesn't need any help getting into the cell or help getting across the basolateral surface. And so as long as we've got a gradient, more water in the lumen, less water in the interstitial fluid, it's going to flow right through that cell and into the interstitial fluid space. And this is the main mechanism via which we move water across our membranes and into our interstitial fluid. Okay. All right. This brings us to a really interesting clinical correlation here. This is called cystic fibrosis. And so cystic fibrosis happens when we don't have the ability to actively pump those solutes to create that solute gradient and to absorb water, okay? And so if we think about the way that we absorb water, it's the same thing that applies when we secrete mucus or any type of water as well. So when we secrete, we also secrete fluids into the lumen of our GI, and that requires the active pumping of solutes, such as chloride here. Oops, chloride here. Okay, my pointer is not cooperating. Okay, so chloride here is being actively pumped from the cell out into the lumen. And this would create a gradient that allows water to then follow, right? Again, creating that gradient where water can follow. This works in both directions for absorption in some areas, but also for important secretion in other areas as well. And so what happens is we've got chloride being pumped out, sodium being pumped out. This creates a gradient out here where water can move naturally to aid in that secretory process. Now, in the case of cystic fibrosis, there is problems with the active pumping of chloride into the lumen. And so what happens is we don't have enough water moving into the lumen to aid in the secretion of mucus. And mucus we know is important in different areas in our body. So for example, in the respiratory area, it's going to be important to secrete mucus into the respiratory airway to clear any debris. It's also going to be important in the reproductive function. So mucus is what helps to move the ovum along the reproductive tract and aid in that, that reproductive process, in the fertilization to create that embryo. And so if we don't have the ability to actively pump chloride into the lumen of these spaces, we don't have the ability to create that gradient where water can move to these spaces and allow the dilution of mucus to be more of a watery consistency. And so in cystic fibrosis patients, we see this very thick, very viscous mucus secretion that is not very effective, right? When the mucus is too thick, it doesn't allow for the clearing of debris and for the movement of particles across the various membranes where we need that mucus secreted. And so some of the clinical consequences in these patients is respiratory infections, so frequent upper respiratory infections because of the ability of this mucus to move adequately because it doesn't have as much water involved in it is much thicker, much more viscous. We also see infertility in these patients, whether it's male or female, because again, of the inability of sperm to be mobilized appropriately, of ovum to be mobilized appropriately. And so those are two of the main clinical consequences that we see in cystic fibrosis patients because of these thicker, more viscous mucus secretions. All right. Okay. And then the final thing that we'll discuss here is called transcytosis. And transcytosis is really a combined process of endocytosis as well as exocytosis. And this is another way that we can move particles across our membranes. So we can simply engulf particles at the apical surface, and we simply just kind of pinch off a part of the apical membrane, engulf that particle, bring it into the cell. This is called endocytosis. And then we can transport it across the cell. And then when it gets to the basolateral surface, we simply sort of evacuate that particle. The membrane of the vesicle will fuse with the membrane of the basolateral surface here. And then we can kind of just spit that molecule or particle out on the basolateral side of the cell. Okay, so transcytosis is another way that we can move molecules through our cells, and it really involves endocytosis on one end and then exocytosis on the alternate end. All right, okay. So we'll spend the remaining minutes here looking at a few practice questions. All right, so I'm going to launch a poll. Okay, final 10 seconds. All right. Okay, majority of you selected B. Which of the following terms describes a fluid that has a higher concentration of total solid particles than another solution to which it is being compared? And so this is correct. Hyperosmotic is the way that we describe a solution that has more solids dissolved, okay? And again, we want to remember here that when we're talking about this osmolarity, we're usually comparing two solutions, right? So hyperosmotic is how we compare the solution that has more solids to a solution that has fewer solids. Okay, let's look at another question here. So again, all right, let's see. So what happens to a cell that is put into a hypertonic solution? Many of you selected C or number 3, and that's correct, right? It will shrink, okay? And again, we want to think about the area that has more water, okay? So we want to think about the area that has more water. So again, if we're putting it into a hypertonic solution, this means that the solution has more solutes, less water. The cell has more water, less solutes, and so water will move from the inside of the cell to the outside of the cell, and so that cell will shrink, okay? Someone's asking me to just go back to this previous question and explain why this is hyperosmotic here again. So we're thinking about describing a fluid that has a higher concentration of total solutes than another solution. So we can think about this as our cell. And so we want to think about the solution out here, the extracellular fluid, versus in here, the intracellular fluid. And if we're saying that there's a higher concentration of solutes out here, right, and a lower concentration of solutes in here, we can then say that the extracellular fluid is hyperosmotic when compared to the intracellular fluid, okay? In other words, it's got more solutes dissolved. This has less solutes dissolved. All right, I hope that makes sense there.
So is it referring to the concentration outside of the cell rather than inside for this, like these types of questions?
So this can actually be, it usually is referring to the extracellular fluid, but this can be any relationship. So we're just talking about any fluid. So if you think about a fluid and we're saying that it has higher concentration than another solution, that's hyperosmotic. So it's usually one fluid relative to another. In this scenario, yes, it's going to be the extracellular fluid relative to the intracellular fluid. And that's the way it usually is, okay? And so we're saying that the solution in question, right, the one that has higher solute concentration, is hyperosmotic when compared to another solution, since there are less particles in that solution. I hope that makes that a little bit clearer. Okay. All right, so we've got one more question here. And then we will call it a day. So one more question. All right, last 10 seconds here. Okay, so in an epithelium specialized for absorption or secretion, one side of the epithelium faces the lumen of a body's cavity. The other side, referred to as the facing membrane, faces the interstitial fluid. Which of the pairs below is the correct association between the two sides of the membrane and the term used to describe them? So a lot of response selecting D, or number 4, and that is correct, right? The side that faces the body lumen is called the apical side of the epithelial membrane or epithelial cell. The side that faces the blood or the blood-facing surface is called the basolateral membrane, okay?